Welcome students to MBBS Online Lecture 2021 YouTube channel. The topic for today's lecture is about immune tolerance and disorders. In those disorders, we will discuss in detail about the hypersensitivity reaction and the autoimmune disease. This is the last topic for the immune section and immunity. So let's discuss it. So first of all, the learning objectives for today's lecture. These are the learning tolerance in case of adaptive immunity. What is tolerance? Vaccination. What is the concept of vaccination? Passive immunity, allergy and hypersensitivity, types of hypersensitivity, and examples of hypersensitivity. The section for the hypersensitivity is fall in the category of immune disease and autoimmune disease. While the passive immunity, the type of immunity, we have given a brief overview of passive immunity in previous lectures. So let's first of all, immune tolerance. What is immune tolerance? Have you ever thought about that why our body just only react to foreign substances and foreign bacteria or viruses, not initiating immune response to our own tissues? This is because the immune tolerance. Tolerance refers to recognition made by our immune cells to distinguish from self antigen from non-self antigen. So this is the immunological memory in our immune cells that are pre-processed in a manner that they distinguish self-antigen from non-self-antigen. Self-antigen are body's own tissues, while the non-self are the virus, any micro or pathogen. Therefore, person's own body tissues are not harmed during immune response. This is the reason why our body does not cause harm to our tissues. Our immune system does not cause harm to our own tissues. Mass tolerance results from clonal selection during pre-processing of immune cells. Pre-processing occurs such as of T lymphocyte in thymus. In thymus, the pre-processing of T lymphocyte occur, which is a secondary lymphoid organ. In pre-processing, where all or most of the clones of lymphocyte that are specific to damage the body's own tissue cells appear to be self-destroyed by apoptosis because of their continual exposure to body's antigen. So what happens in the thymus? Our body or the thymus tissue that is a lymphoid organ, it basically exposes foreign and and body's antigen to these newly formed and newly developed cells. Those are T lymphocytes or B lymphocytes. So if the T lymphocytes and B lymphocytes or these leukocytes are initiating or causing damage to our own body cells that are our self-antigen. So these are self-destroyed by apoptosis. This is the case in 95% of the cells are destroyed by apoptosis. Only 5% are the cells that are harmless to our own body tissues. So failure of immune tolerance causes autoimmune disease. So what are the autoimmune diseases? Here is a brief discussion for this. Rheumatic fever. In this type of disease, body become immunized against tissues in the joints and heart, especially the heart valves, after exposure to a specific type of streptococcal toxin that has an epitope structure similar to the structure of body's own self-tissue antigen. Here is a conceptual thing that there is a type of bacterial toxin that has an epitope, that has an antigen that has epitope structure similar to the structure of body's own self-tissue antigen. So what happens to our immune cells? They cross react. Instead of reacting to a streptococcal toxin, they react to our own body cells, causing rheumatic fever, that is particularly in joints and heart. Epitope is a structure that binds and that is recognized by the antibody. So, glomerulonephritis. In this person become immunized to basement membrane of glomeruli. The basement membrane of glomeruli is attacked by our immune cells in causing inflammation in glomeruli capsule. Myasthenia gravis, immunity develop against the acetylcholine receptor of the neuromuscular junction causing paralysis. Multiple sclerosis in which immune cell attacks myelin sheath or causing disrupting nervous communication. Systemic lupus erythematosus, this causing immunity to many different tissues at a time, can cause excessive damage or even death. So many multi-organ failure can be result of the systematic lupus erythematosus. So this is a myasthenia gravis. Here you can see the normal neuromuscular junction. These are the acetylcholine, and these are the acetylcholine receptors which are in on the surface of the muscles. When these receptors bind to the when these acetylcholine molecules bind to the receptor, it causes action potential to generate in the muscles. While in myasthenia gravis, what happens? Our immune cells or our antibodies react against those receptors that are nicotine or acetylcholine receptors. So destroying these receptors, causing no action potential to generate in the muscles. While in SLE, systematic lupus erythematosus, there is a formation of widespread antibody that react against our own body cells, causing multi-organ failure. Rheumatic fever, there is a streptococcal that is similar to our antigen that is present on heart valves or joints, causing B cell and T cell to cross react our body on tissue cells, causing rheumatic fever. So, second is the immunization. What is vaccination? What is the concept of vaccination? It is a process that can be used to produce acquired immunity against specific disease. So, as we have discussed, acquired immunity is developed within person's life and is not present at the time of birth. So, acquired immunity can be artificially produced in a person for a specific disease. This can be done by injecting dead organism, can be used to protect typhoid fever, whooping cough, diphtheria, and other bacterial disease. Injecting dead organism also possesses those antigen that can be recognized by immune cells and causing immune response in generating memory cells. Destroying toxic nature, making them harmless. This can be immunization can be done for tetanus, botulism, and other toxic disease. Injecting live organism as a case in smallpox, yellow fever, poliomyelitis, measles, and other viral disease such as nowadays coronavirus. Vaccination is also works on these three mechanisms. Passive immunity. Thus far, all the acquired immunity I've discussed have been active immunity. Active acquired immunity. That is a type of we have discussed active acquired immunity. Now that is the person's own body develops antibodies or activated T cells in response to invasion of the body by a foreign antigen. So when a naturally foreign antigen accumulate or invade in our body, our body cell develop T cells and B cell and immune and antibodies against that foreign antigen. This is the case in active immunity. While temporary immunity can be achieved in a person without injecting any antigen, this can this temporary immunity is achieved by infusing antibodies, activated T cells, or both obtained from the blood of someone else or from the animal that has been actively immunized against the antigen. So what happens in passive immunity? Basically, the artificially made antibodies, activated T cells, helper T cells particularly, are obtained from someone else, or they are cultured in some animals such as in animals that have been extracted and infused in our bodies, providing temporary immunity. Antibody lasts in the body for recipient 2 to 3 weeks, and during that time, the person is protected against the invading disease. Activated T cell lasts for few weeks if transferred from another person, but only for a few hours to few days if transferred from an animal. Such transfusion of antibody or T lymphocyte to confer immunity is called passive immunity. So passive immunity is basically the transfer of or infusion of antibodies, activated T cells that can be obtained from someone else's blood or can be some other sources such as from animals that have been actively immunized against that particular antigen. So this is also the for the specific antigen. Let's move towards the hypersensitivity. This is a disorder of immune system that is a case in autoimmunity. It is injurious immune response are grouped under hypersensitivity, as we have discussed. What is it? It is an exaggerated immune response to harmless antigen or often environmental antigen that is often related with inflammation and causing damage to body's own cells. So this is a exaggerated hyper hyper immune response. We say increased immune response to a harmless antigen that is not harmless to our body or often environmental antigens such as pollen grains can which result in the of inflammation of particular site causing damage to our own body cells or tissues. Prolonged immune response is also cause of chronic inflammation. What are the causes for hypersensitivity? This is the autoimmunity reaction against self-antigens. Failure of cell tolerance in body can cause autoimmunity. This is the first case for the hypersensitivity. Reaction against microbes. So reaction, the particular initiation of immune response against any microbe can trigger inflammation. That inflammation can result in the severe damage to our body cells. Such as if antibodies are produced against such antigens, the antibody may bind to the microbial antigens to produce immune complexes which deposit in tissue and trigger inflammation. The T cell response against persistent microbes may give rise to severe inflammation, sometimes with the formation of granulomas or granulomatous inflammation. Rarely, antibody or T cell reactive with a microbe cross reacts with host cell tissue, as a case in rheumatic fever. Reaction against environmental antigen. Some environmental antigens such as pollen, animal dander, and etc. can trigger excessive immune response that can cause injury to cell. It is termed as allergy. So allergy is basically the hypersensitivity that is the hyper response of our bodies or immune system to environmental antigen that is not harmful to our body, causing injury to our cells. So what are the types for the hypersensitivity? These are divided into two: antibody mediated and cell mediated. What are the types for antibody mediated? These are immediate type 1 hypersensitivity, antibody mediated type 2 hypersensitivity, immune complex mediated type 3 hypersensitivity, while cell mediated is cell mediated type 4 hypersensitivity. So type 1 hypersensitivity, often called allergy, injury is caused by T helper 2 cells, IgE antibodies, and mast cells and other leukocytes. So these are the culprits for the type 1 hypersensitivity. Mast cell release mediator that act on blood vessel and smooth muscle as well as cytokines that recruit and activate inflammatory cells. What are the chemical mediators that are released by mast cells that causes inflammation in our cells? These are the vasoactive amines, particularly histamine, that causes vasodilation, increased vascular permeability, smooth muscle contraction, and increased secretion of mucus. As you can see, these are the stages and these are the stages for the inflammation. And lipid mediators, these are prostaglandins and leukotrienes. Prostaglandin PGD2 is the most abundant mediator generated by cyclooxygenase pathway in mast cells. As you know, there are two types of pathway by which these prostaglandin leukotrienes are produced. These are the cyclooxygenase pathway and lipoxygenase pathway. It causes intense bronchospasm as well as increased mucus secretion. The leukotrienes CTC4, LTC4, and LTD4 are the most potent vasoactive and spasmogenic agent known. They are most potent in histamine causing vascular permeability. LTB4, as we have discussed, that these are the chemotactic factor. Cytokines that are released that cause recruitment and activation of macrophages. These are tissue necrotic factor and chemokines that are interleukin 4, interleukin 5 that has an effect on other immune cells. So here you can see what happens in this picture. Allergy, allergen that is called pollen. Pollen comes in our body. It is sensed by B cells or the T helper 2 cells. Activation of T helper 2 cells and IgE class switching in B cells. What happens? T helper 2 cells sense these antigens by dendritic cells. Then it presents these cells to B. Then it activates B cell. Activating B cell produces IgE type of antibodies, such as in this case. These IgE antibodies bind to the FCERI receptor that are present on the mast cell. As there are two types of region on the antibody. The first one, that is a variable antigen, is also called FAB region, and the constant region is also called FC region. So the FC region of the antibody bind to the FCERI receptor present on the mast cells. These are the FCRI receptor. They bind to it, and we say that these are sensitized. So what happened on repeating exposure to allergen, and second time the allergen is exposed to these antibodies or T cells, it causes mast cell to release many granules and chemical mediators. These contains vasoactive amines, as we have discussed, and cytokines that can cause inflammation and trigger infiltration of neutrophils and macrophages. So there are two types of reaction. These are the late phase reaction and immediate response. What are the immediate response after granulation? These granules contain histamine, protease, chemotactic factors, while arachidonic acid, these are prostaglandin leukotrienes. These causes vasodilation, vascular leakage, smooth muscle spasm. These are the chemicals that are involved in causing immediate response, while secreted cytokines, these causes late phase reaction, such as interleukin 4, tumor necrotic, tissue necrotic factor. This causes leukocyte infiltration, epithelial damage, and bronchospasm. So let's discuss what are the clinical examples and daily life examples for the hypersensitivity type 1 reaction. First one is an anaphylactic shock, that is fall in blood pressure, shock caused by vascular dilation, airway obstruction due to bronchial spasm caused by prostaglandins due to laryngeal edema. Second is a bronchial asthma, airway obstruction caused by bronchial smooth muscle hyperactivity, inflammation and tissue injury caused by late phase reaction in the bronchial tissues, hay fever or allergy, increased mucus secretion from the upper part of airway, inflammation of upper airways and sinuses. That is the sign is a runny nose and redness in the nose. Food allergies. Sometimes some people are allergic to peanut butter. This is the most common food that is allergen. So many food can cause allergy to some people, causing excessive peristalsis due to constriction of intestinal muscle, resulting in vomiting and diarrhea. So let's discuss type 2 hypersensitivity. It is antibody mediated type 2 hypersensitivity disorder. These are caused by antibodies that bind to fixed tissue or cell surface antigens, promoting phagocytosis and destruction of coated cells, opsonized cells. Coating of a bacteria or any cells by antibodies is called opsonization. Or this trigger triggering pathologic inflammation in tissues. So what happens in type 2 hypersensitivity? Antibodies are bind to the surface of any cell or surface antigen, causing promoting phagocytosis and inflammation in tissues. What is the mechanism? First one is opsonization and phagocytation. This is done by complement product, that is C3b, as we have discussed in the previous lecture of complement system. C3b is a product released by complement product that causes complement system that causes opsonization and phagocytosis. Inflammation. Complement system by classical pathway releases some products such as C5a, C3a, C4a. C5a is a chemotactic factor. C3a, C4a, these recruits neutrophils and monocyte, triggering inflammation in tissues. This is an again mechanism of hypersensitivity type 2. So inflammation is the second mechanism. Third one is the antibody mediated cellular dysfunction. So what happens in this? Antibodies are directed against host protein, impairing or dysregulate important function, such as in myasthenia gravis. So what happens? Antibodies are directed to host cells. This causes destruction and dysregulation of important functions of some of that tissues. Such cases myasthenia gravis, in which acetylcholine receptors are dysregulated or destroyed. First one, that is opsonization. So this is the cell, opsonized cell. Here the complement system, a complement product C3b causes antibody C3b receptor, such as antibodies against that cell, causing phagocyte to recognize this cell and cause phagocytosis of this cell. While complement and Fc receptor mediated inflammation, second type mechanism, that is Fc receptor present on these cells, causes complement activation. Such products C5 and C3a cause a recruitment of neutrophils and macrophages that cause inflammation and tissue injury. Now, third one, that is antibody-mediated cellular dysfunction. In this, antibodies are directed against those tissue cells or tissues or cells that cause dysregulation of function, such as in this, the TSH thyroid stimulating hormone receptors are destroyed by antibodies, causing hyperthyroidism. While in this, the neuromuscular junction, antibodies are directed against SCH receptor, acetylcholine receptors, causing persistent paralysis in the muscles. So what are the clinical manifestations for hypersensitivity type 2? These are disease. What are the target antigens to which antibodies are directed? What are the mechanism by which antibodies works and by which hypersensitivity type 2 works? What are the clinical pathological manifestations? First one is autoimmune hemolytic anemia. Target cells are red cell membrane proteins. By which mechanism? These are the opsonization and phagocytosis of RBCs, causing hemolytic anemia, as the case seen in erythroblastosis fetalis, hemolytic disease of newborn. Also, the sensitized mother causes production of antibodies to fetus cells, Rh positive cells. Second is autoimmune thrombocytopenia, purpura. The target antigens are platelet membrane proteins such as glycoprotein 2b and 3a integrin, causes caused by opsonization and phagocytosis of platelets, increased bleeding. Acute rheumatic fever, streptococcal cell wall antigen, antibody cross-react with myocardial antigen. This is also, we have discussed rheumatic fever, in which streptococcal cell wall antigen and our myocardial antigen has the same epitope. Therefore, antibody cross-react with each other and causing damage to heart valves. Mechanism inflammation, macrophage activation, causes myocarditis, inflammation in heart. Myasthenia gravis, what is the target antigen? These are acetylcholine. Antibody inhibits acetylcholine binding, down regulates receptors. So muscle weakness and paralysis is the clinical pathological manifestation. Graves' disease, hyperthyroidism. In this, the antibodies are targeted on TSH receptors. Antibody mediated stimulation of TSH receptors. This is the mechanism. What it causes? It causes hyperthyroidism. Pernicious anemia. We have discussed in the anemia chapter. It is a type of anemia in which intrinsic factor of gastric parietal cells are destroyed that causes less release of intrinsic factor and absorption of B12 from the proximal duodenum. So what is the mechanism? Neutralization of intrinsic factor, decreased absorption of vitamin B12. It causes abnormal myelopoiesis anemia, mean such as megaloblastic anemia. So another type of hypersensitivity. In this antigen antibody complexes are found in the circulation with deposit in the blood vessel leading to complement activation in acute inflammation. So what happened in this type of hypersensitivity? Antigen and antibody complexes are found in response to exposure to foreign antigen. These within the circulation is seen in this picture. There are the free antigens. B cells recognize these antigen, causes differentiate into plasma cells, causing release of free antibodies into the circulation inside a vessel. These antibodies form antibody antigen complex, such as seen in this picture. These complexes are deposited on the walls of endothelium. This can trigger the infiltration of neutrophils or complement activation, causing vasculitis or vessel damage. So, what are the clinical manifestation? First one is a systemic lupus erythematosus. We have discussed SLE, that in which multiple organs are or multiple sites are attacked by immune complexes. In this, nuclear antigens are involved. What are the manifestations? These are nephritis, skin lesion, arthritis, and other. Post-streptococcal glomerulonephritis. In this, the immune complex is deposited in the walls of glomerular basement membrane, causing nephritis. Reactive arthritis, bacterial antigen. The complex is formed with this type of antigen. It deposits in causing acute arthritis. Serum sickness, various proteins such as foreign serum proteins such as in horse, anti-thymocyte globulin. This causes arthritis, vasculitis, nephritis. Arthus reaction, various foreign antigens are involved that can cause cutaneous vasculitis. So type 4 hypersensitivity. This type of hypersensitivity is mediated by T cell, as we have discussed. This type of hypersensitivity is T cell mediated hypersensitivity. So two cells are involved. These are cytokine mediated inflammation. Sorry, there are two type of T cell mediated reactions. These are cytokine mediated inflammation that are caused by T helper CD4 plus T helper cells and direct cell type cytotoxicity. This is caused by cytotoxic T cell CD8 plus. CD4 mediated we will discuss first. In this, cytokine are produced by the T cells, induces inflammation that may be chronic and destructive. It is called delayed type hypersensitivity. Chemical mediators mostly released by T helper 1 and T helper 17. These are gamma interferon and interleukins. So we have discussed that T helper cells are the primary cells to respond against any antigen. These are the cells that are involved in activation of many immune cells such as B cells and T cells. So T helper cells are the most important. So these release many chemical mediators. These are released by T helper 1 and T helper 17. Such mediators are gamma interferon and interleukins. This can cause inflammation, such as seen in this diagram. Cytokine mediated inflammation, APC representing tissue present antigen to the CD4 plus. This release cytokines that causes inflammation. These cytokines may be interleukin 4, 5, 1, 1, 2 that are that causes infiltration of neutrophils and B cell proliferation differentiation, or gamma interferon. This causes macrophage activation, can cause tissue injury. So type 4 CD8 plus mediated, second type 4 hypersensitivity. In this form of T cell mediated tissue injury, CD8 plus cytotoxic leukocytes kill antigen bearing target cell. So CD8 plus, we have discussed the function of CD8 plus cytotoxic T cells. They directly bind to the antigen bearing cells and kill them by releasing many perforin enzyme or enzyme into their cytoplasm, such as cell killing and tissue injury occurs. What are the clinical examples? First one is rheumatoid arthritis. Specificity of pathogenic T cell, collagen citrulline cells protein, part of the mechanism, inflammation mediated by T helper 17 and T helper 1, cytokine, and role of antibody mediated, and immune complexes. What are the manifestation clinical? Chronic arthritis with inflammation, destruction of articular cartilage and bone. Multiple sclerosis, as we have discussed previously, this is the attack by our immune system on the myelin sheath. So, what are the specificity of pathogenic T cells? Protein antigen in myelin. So, inflammation mediated by T helper 1 and T helper 17. Demyelination in CNS, perivascular inflammation, paralysis, and occlusive lesions. So, third one is diabetes mellitus. In this, antigens of pancreatic islets, beta cells of islet or Langerhans, T cell mediated inflammation, destruction of islet cells by CTLs, insulitis. Contact sensitivity, various environmental chemicals such as from IV poison can cause inflammation mediated by T helper 1 and T helper 17 cytokines. Epidermal necrosis, dermal inflammation with skin rash and blisters. So that's the end of the lecture. As we have briefly discussed the hypersensitivity reactions and what is the immunological memory. So that's the end of the immune section and immunity. If you like this video, share it with your friends and subscribe the channel. Thank you for watching. We'll see you in next video.